Welcome back, everyone. I hope you enjoyed some of the fabulous San Diego weather there. I can appreciate it, especially being from Chicago this past winter. We're moving on now to emphasize the interdisciplinary nature of research in neuroscience and the other subjects that have been mentioned today. And near and dear to my heart, Dr. Shu Chien, who is celebrating his 16th anniversary here at UCSD, is an engineer and has focused research on bioengineering. Dr. Shu Chien received an award from President Obama for the research and focuses research on the physics and the interactions and pressures and the biological response in cells. And I'm very happy, and I'm sure you will all be happy to hear about the important research being done by Dr. Chien's lab.
Thank you very much, Steve. Good afternoon. It's been a wonderful meeting so far. I've heard the marvelous talks by Bob Dines, Larry Goldstein, and Nick Spitzer, bringing out the importance of interdisciplinary research. I think Nick said several times you want to break down the silos between engineering and medicine. That sort of really set it up for my talk. And Larry Smart, who's going to finish the talk session today, he's been doing that really very actively, including on himself, personalized medicine with engineering. So this is really a wonderful time for us to get together to talk about how to how to catch the next big wave. So I'm in the departments of bioengineering and medicine, and also the Institute of Engineering Medicine at UCSD. What I'm going to talk about is not what I have done in pressure flow effects on endothelial cells, but rather to talk about the wonderful work my colleagues have done in the Institute of Engineering Medicine and others who can illustrate how engineering can be applied to medicine to improve healthcare that would benefit people. Where the big wave was made at the turn of the century when the human genome was sequenced, and that opened up a lot of new vista for us to follow. And with that, we have so much data, and it's accumulating more and more all the time, and we need to be able to integrate this kind of information. And the integration here, we need the integration of biology, medicine, and engineering. Of course, other sciences, chemistry, math, and other sciences, and to integrate across different levels in the biological hierarchy, from genes to cells to tissues, organs, to the whole body, and even the society. And then we need to integrate education, research, and also the technology and translation. So we have to work with industry, and we have to work with our clinical colleagues for the translation of our research results to benefit people. Talk about the biological hierarchy. As I mentioned, we can go from genes to molecules to cells, tissues, organs, and systems. And even the systems and the genes are related because genes can be studied by using system biology, and what happens in the system can be studied by manipulating the genetic makeup. And I'll start by talking about the nanotechnology, which Nick already referred to several times in relation to neuroscience. And here I'd like to talk about the work that has been done by my colleague in UCSD. Is there another pointer somewhere?
Should be right on the podium.
Right on the podium? Oh, here it is. Thank you. in delivering nanoparticles with drug payloads, the one thing we have to deal with is that these are usually foreign to the body. They are recognized as being foreign, and they cannot do what we would like them to do. So what Liang-Fang has done is to use the native cell, the red blood cells. You and I have a lot of them floating around, and what he did was to lyse them to take out the content and use only the membrane, which then has various kinds of proteins and lipids in there, but without the interior in which you can package the nanoparticles. In this way, you can deliver the nanoparticles to various tissues and cells and do their job. It's a camouflage nanoparticles so that the body will not reject that. You can deliver therapeutic agents, deliver imaging agents, and also you can use it to absorb toxins by using the surface of the membrane, because the rest of the membrane can do that. So this is a very innovative approach that make us possible to use the nanoparticles much more effectively without many of the side problems. Besides the red cell membrane, he has also used the cancer cell membrane. Here's a cancer cell which has been lysed, so you have only the membrane without the interior, without the nucleus with the genes and all the proteins in the cytoplasm, but only the membrane. In the membrane, we can package the nanoparticles in there as the core, and this is the cancer cell-coated nanoparticles, CCMPs, and these can be then used to deliver antigen to antigen-presenting cells to work on immunological approaches, and also it can then use these to link with the cancer surface molecules. We call it homotypic, that is, same molecules will recognize each other, so we can deliver the payload of treatment particles to the cancer cells by conjugating them with the same surface molecules, so they can get specifically to the tumor cells. One of the important things for treatment is not to get it to the regions that are not desired. We want to get them to the region we want, that is the cancer in this case. So this will really allow us to do very specific presentation of the therapeutic agents. This is the work done by another colleague, Ada Amatari, and what she has done is also on nanoparticles, using these hydrated polymer nanoparticles, in which we have confined water encapsulated payload within this polymer nanoparticles. And then you can irradiate the nanoparticle with infrared radiation, which will have heat transfer to the polymer matrix, and this photothermal heating will make the polymer become rubbery and release the water. So water becomes free, and then the payload can be delivered. So that this is a target delivery. You can shine the infrared at desired location, so you can deliver the agent where you want it. So these two are, now this method is not only good for nanoparticles for cancer treatment, but it can be used for a variety of other kinds of applications by using photoactivation control, the chemistry in time and the space. I'd like to move up to the tissue level. Tissue engineering is an important topic. We deal with cell interaction with the matrix on which they sit, and interaction between cells and cells. And Larry has given an outstanding talk on stem cells. I'll just show one slide. Of course, you know that stem cells, pluripotent stem cells, can be differentiated into a variety of cells, such as blood vessel, heart, liver, and pancreas. And it's well known that the differentiation and the proliferation of the cells depend on its microenvironment. That is the physical-chemical properties of the environment. There is a lot of study on the chemical stimuli, such as growth factors and so forth, on the signaling pathway to cause the stem cells to either differentiate or proliferate. And it's also known that extracellular matrix, the proteins here, such as collagen, fibronectin, etc., they interact with the cell to modulate their fate, as well as the cell-cell interaction. What has been shown in recent years is that not only the chemical factors, but also physical factors, such as mechanical stimuli, such as pressure and flow, can also modulate the behavior of the stem cells. And furthermore, the matrix rigidity on which they sit has a very important influence on what the stem cells do. So I'll just show a couple of slides on these two factors, the rigidity effect and the extracellular protein effect. The rigidity effect was discovered by Adam Angler in our department when he was at UPenn as a graduate student. His thesis work showed that if you modify the rigidity of the environment of the stem cell, you can get different kinds of differentiation. And for example, use a very soft matrix, then the cells that has multiple potentials will go into the soft cells, which is the neuron. And you can see this is the markers for the neurons. You can see they show up, but not the muscle or the bone. On the other hand, if you use intermediate matrix, then you see the muscle differentiation, not the nerve nor the bone. Now, if you use a very hard environment, that is, you can see it's more than tenfold over this soft one in rigidity. Now you get bone differentiation rather than the muscle or the neurons. So the cells can sense their surrounding. What the rigidity is, it will become similar to that surrounding. So there is a continuous interaction between the cell and its microenvironment. The cell can be modified by its environment. In order to understand what kind of matrix proteins can modulate the fate of the stem cell, we have a microarray system. You can see there are many, many of these spots on which we can spot different kinds of matrix proteins. And then we put the stem cells on there to see which matrix protein and their combinations are most beneficial for a particular kind of differentiation that we want, or proliferation. Then we can decide with this relatively high throughput technology what is the optimum environment for the stem cell, either for differentiation or proliferation. This is a higher magnification, and then we get this kind of there's a spotting device, make these kind of array. Then we can look at the array and their stand, so you can get the output from which you can determine the optimum ECM composition, not just one protein at a time, but various combinatorial ways. What are the 2 or 3 combinations that are best for the differentiation? So this will help us to make a large quantity of stem cells and also to direct their differentiation into a particular lineage. This kind of approach we have adopted to use it to determine the anti-cancer drug, because as you know, cancers don't always respond to one kind of drug, but very often they respond more effectively to combinatorial drugs. And in order to study that, it takes a lot of iterations, a lot of experiments, but using this kind of approach, you can spot different kinds of drugs in different combinations. You can put a patient's cancer cells on there to see which one surrounding which kind of drug is most effective for this kind of cell. Now let's move up to the function level, at the organ level, and this is a slide from Todd Coleman in our bioengineering department, and this is the epidermal electronics. He is able to use a very thin piece of material put on the skin. As you can see, it's very thin and very flexible. You squeeze with the fingers, you can see it will deform, but without changing its structural features. You can still function. It's ultra thin, ultra light, ultra low modulus, very soft, and this kind of the electronics are robust to mechanical deformations. So you can use this to study various kinds of signals from the organ or tissue that we're looking at. And in there are sensors for electrical stimuli, for temperature, for mechanical strain, and for all kinds of sensing, and also has the LED system in there, wireless communication, and and light sensors, so that it's all embedded in this thin piece of, small piece of sensor, and you can put it on the abdomen of the pregnant mother and record ECG, electrocardiogram from that. Instead of like in the old days, you have to strap up the mother and with all kinds of restraining devices. Now just one piece of this kind of tape-like thing would do it all. And the same is true for the infants. Instead of having the infant strapped up like this, now you just have this little piece here, and you can record all the things you want, non-invasively and very effectively, and this is very, very useful. Now, Nick Spitzer talked a lot about the brain, and I'd just like to show this one case that we can use engineering approach to sort out the brain waves, the electroencephalograph. It originates from different parts of the brain, and they're adjacent to each other, so their waves interfere with each other. So when you record here, it is affected by activity here and there, and there is a way using engineering technique, mathematical modeling, to sort out the signal in the face of many noises. So here is an example of the so-called cocktail party problem using this blind source separation to sort out the noises from the signal. So people are there maybe talking and having various kinds of noises like music of various kinds, and this person is talking, it cannot be heard. But if you sort out these, then you can hear what he's saying. Then people appreciate it and will applaud. So this is a sort of example showing how the brain waves can be sorted out by using this kind of computational approach, so you can get what you want, taking away the noises. And this is so you can get separation of the brain from the non-brain signals, so that this kind of electroencephalograph can be analyzed with more meanings. Now we move up to the systems level. The system biology is a very rapidly developing field in recent years. There's a slide from my colleague Bernard Paulson showing how the system biology brings together biological sciences and system sciences. So from cells to genes, and here your system approach from modeling to mathematical solution. So you bring the two together to have a genome scale interaction network. So you understand how the genes behave in the presence of other genes and other signals. Now, as I mentioned, the turn of the century, we sequenced the human genome, but in 2007, Jim Watson decided to sequence his own genome, and that paid about $2 million. And you saw Craig Venter's picture earlier, and Craig did it himself also, but those are very expensive endeavors, and Science magazine every year selects a discovery of the year or molecule of the year. Then in 2007, the selection was human genetic variation. In other words, instead of having a group, you look at individuals. These genomic variations can make a difference in disease susceptibility responses to drugs. So how we are susceptible to diabetes or hypertension or certain kind of cancer could be obtained. The information could be obtained from the genomic sequence, and also our responsiveness to drugs, whether we're hypersensitive, having side effects, or not sensitive, or just perfectly sensitive, can be determined this way. So it's very important. So everybody wants to know not just the human genome, but my genome. And the target at that time was to have a $1,000 human genome determination of the individuals from the $2 million that you saw. And in 2001, actually, it was very expensive, and 208 it drops down. In 2013, last year, to 6,000. But Illumina has really led the way in doing this, and they said now it's already below 1,000. It's reached the target, so that we can all get our human genome sequenced if we're willing to pay that, or maybe one day the insurance will do that. But there are also other kind of questions like if you know all of these, is it good or bad? Just like sickle cell disease, when it was first decided to do the gene sequencing, gene detection of sickle cell disease, there was a lot of argument whether you should do that or not. Should the man and woman with sickle cell trait, should they marry or not? You know, all of these effects. If we know too much about ourselves, there are other consequences. So it's not just medicine. There is humanities and social science considerations as well. Now, the genes are very complex. This slide taken from Trey Ideker shows the gene circuitry. The genes are connecting with each other almost like a modern art. And not only the genes, the DNA sequence, the ACTG sequence, but also the epigenetic modifications outside the sequence. They can be modified either by changing the histone around which the DNA wrap. If you change the histone, then this will change the gene expression. And the other is the DNA methylation. If you methylate the DNA at certain spots, you can change the gene expression. And finally, the microRNA, which was which became popular in the last decade, it is another kind of epigenetic effect. So besides the genes themselves, the sequence themselves, the epigenetic factors need to be considered. So we really need a system biology approach to bring all of these together in a meaningful way. So the data is going to be huge, and I'm sure Larry will be able to talk to this much more effectively and knowledgeably than I do, but I'd just like to bring up the big data is a big deal. It's really very, very important because we're getting data left and right, so we need to be able to structure them such as for molecular profiling, phenotyping, for imaging, and so like imaging here, the sequencing here. We need to be able to handle the data using the big data approach. And this is not only important for us in healthcare to capture, curate, store, search, share, transfer, analyze, and visualize data, but this is also can be applied to business, to legal actions, to traffic analysis, and determination of research quality, finally diagnosis, treatment, and prevention of disease. So this big data is really very, very huge in its implications. Now, I remember days I thought terabytes were huge. I mean, the supercomputer center handles that. Our lab has nothing close to that, but that's small now. In 2012, 2.5 exabytes data were generated every day. Exabyte is here. So if it's every day, then a year is already got to the zettabyte, and that's 2012. Today is 2014. I'm sure it's already, Larry will know, whether it's yottabyte. Then what's the next name? I don't even know, but maybe it's invented already. If not, it has to be. But anyhow, we need to deal with this big data, and then need engineering and medicine to work together. Now I'd like to show some kind of integrative results. This is to apply engineering modeling to cardiovascular surgery. This is a vascular anomaly, and we can do the model simulation to see how the flow will work. And here's the X-ray showing the actual operating room picture. Then we can do simulation and even trials of operation without the real operation. We can do the simulation and see what's the best way, what the flow will be like. So this will be good. And surgery is going into robotic surgery, non-invasive, minimally invasive surgery, going through the natural orifices, so we minimize the operation wounds. And There are other ways of using engineering approaches to improve the accuracy of operation, to remove like a tumor as much as we can without damaging the nerves. In this picture, this is the work done by Shu Chien's group, and together the surgeons in UCSD, they can put in dyes to stain the tumors and then stain the nerve, because it's very important not to injure the nerve. Like a prostate cancer operation, if you damage the nerve, you have all kinds of problems with urination and sexual action and so forth. So it's very important to preserve the nerve. So the way to do it is to be able to stain both of them. And Roger developed this kind of stain to stain the tumor with this ACPP and pseudo-colored green, even though the fluorescence was not green originally, but he's like a magician. He can make it green. And then he can make the nerve also target stained and make it aqua. So you have the nerve stained and the tumor stained. You can switch back and forth to look at the two, so you can remove as much tumor as you can without touching the nerve. I mean, that achieves the two major purposes at once. Now, engineering has helped us in many ways. Here again, we go to a neuro-related problem, and this is the work done at Medtronic. Paul Citron is our adjunct professor, and this is a slide showing how electrical stimulation can be used to treat something like the Parkinson's disease. Here is a patient, and this has sound. They're talking about Swedish, so whether you can hear it or not doesn't matter, but it does have the screen, on the screen, the English subtitle. But so that, oh, sorry, I should have clicked on here. So this is a pianist, very famous. Can you hear it? It's not too loud, but it's okay. So you just look at the title, subtitle. So he was shaking like this, but then they had an implanted electrode in his brain. Oh, now we hear the sound, yeah. I think it's actually Danish. I think some of you may understand that. So when you stimulate him, he can pour coffee, no shaking. So if you put in the right place, it's not easy to put in the right place. So in December that year, he showed in front of the audience and played his piano. He was not able to do it without this. You see, the patients were the audience was really moved. You see, that's really what science is all about. You can really do so much for the human being, for our health and happiness. And finally, I'd just like to mention that information technology, wireless health is playing a very important part in medicine, healthcare, and that's, I'm sure, Larry is going to talk about, so I'll just show this slide and without saying anything more, but this is very important. So to summarize, the frontier areas that I mentioned so far include nanoscience and nanotechnology, imaging, genomics, regenerative medicine, system biology, novel surgery and devices, wireless health, and personalized medicine. And overriding principles that guide all of these that we must not forget is that we need to be integrative and interdisciplinary, and we go across multiple scales. We must translate our innovative research to clinical application to benefit a patient. And we have to have low-cost effectiveness. Otherwise, everything just gets more and more expensive. We cannot afford that. So the engineers have a responsibility to develop new ways to better diagnose, treatment, and prevention, but with less cost. And equitable distribution of health care to regions that are not as affluent as where we are, and to be able to get this kind of benefit. And finally, we must have integrity and ethics. That goes beyond everything. It must be there. So the healthcare is undergoing tremendous transformation. It's instead of just being in the hospitals, it can be in multiple locations because the wireless approach, you can go to many places, and instead of having fixed appointments, you can do it various times, and instead of treating everybody as part of a population, you can also begin to look at individuals with these personalized approaches. The healthcare professionals, instead of reacting to disease, they will focus on prevention. And the surgeries, instead of being invasive, will become non-invasive. The cost, we like to see, instead of being expensive, but cost-effective. So in summary, it's individualized preventative medicine that is less infrastructure dependent and more patient-oriented and cost-effective. So I'd just like to show this one slide about the Institute of Engineering and Medicine, which I mentioned at the beginning. The IEM, our discoveries are translated to the clinical side through the collaboration of the Altman Clinical Translational Research Center, as well as the various hospitals, and to industry through the collaboration with Biocom Connect and many other organizations here. We're very fortunate in San Diego that we have these organizations that will foster the translation. And then at UCSD, we have the Von Liebig Center for Entrepreneurism, and of course the Tech Transfer Office, the TTO, which is organizing this symposium. I'd like to thank all of them. And so in summary, not only academia has to work hard on these problems, we need to work with medicine and work with industry, and we need to have the society support, the government, and even international. So with all of these, then we can improve people's health and combat disease. And with all of us working together, we can really catch the big wave. Actually, my thinking is not just to catch the big wave, that's passive. We will make the big wave. That's what we do. We'll make, thank you.